In the first part of the class, we've been discussing thermodynamics, we've been discussing the properties of water and acid-base equilibria and how this affects the protonation state of different biomolecules. We've been discussing amino acids and how these can polymerize into polypeptides, and then how polypeptides, as they get larger, can start to fold and form these three-dimensional structures, which we formally call as proteins. Now, we're going to start putting all this together to look at different chemical reactions that proteins can perform. And so in this way, we're starting to introduce the concept of structure, function relationship. And what we're looking for is how does the structure of the protein and how does that affect the different jobs that these proteins do within the cell that are important for cellular life. So what we're going to look at is we're going to look at, as a case study, two different proteins which at first glance seem to do exactly the same thing. So one of these is called myoglobin and the other one is hemoglobin. They both bind dioxygen, or O2, shown here. Very famous molecule that we know is very important for life on Earth. And so these bind this molecule, but they have different functions. So for instance, we know that when we inhale air and along with it oxygen, which is important. And then that gets delivered to cells throughout our body. Now, that doesn't happen just by diffusion of oxygen to these other cells. This gets carried through the blood. And the important protein, the protein that's important for carrying this is, of course, hemoglobin. And so O2 uptake happens in the lungs, as seen in the slide on the right. This is delivered to different tissues where O2 is now released. And then this returns back, and then this this gets more O2, and then this is the blood circulation system. When O2 is released to the tissue, myoglobin sequesters the O2, so it binds the O2 and uses, and in this way myoglobin is sort of an O2 storage protein that holds on to the O2 until it's needed for some cellular process. And so how do these two proteins differ? So we know that myoglobin is able to bind to the O2, and hemoglobin has to release it at very certain times. So these both have different properties in when they bind O2 and when they release it. And so these are O2 binding curves, and we'll talk more about this as we progress through the lecture. And so in the black trace, the black data points are what are called an O2 binding curve for myoglobin. And so the important part is that at really low O2 partial pressures, myoglobin is completely filled with oxygen. So all the myoglobin in a cell is completely filled very quickly at low pressures of O2. Whereas hemoglobin, it has a different binding affinity curve, as you can see in this red trace. And so the question is, so that's useful for the varying functions of hemoglobin and myoglobin. While myoglobin, the O2 concentration within the cell is very low, so myoglobin has to be able to bind O2 very tightly, even at really low O2 concentrations. Whereas hemoglobin, again, as a delivery protein, or as a carrying protein and also delivering protein, needs to be able to release this O2 at different concentrations and bind at high O2 concentrations. And so we'll discuss this more later, but the important thing is that they have different functions, and the activities that we can see from the O2 binding curves reflect these different functions. So, as I said, we're looking at structure-function relationship. What causes these different binding affinities for O2 in these two proteins? And the answer is, of course, the structure. And if we look at the structure very quickly, so these are X-ray, these are structures obtained from X-ray crystallography of both myoglobin here on the left and hemoglobin on the right. And so what's interesting is that we have what's called a ligand binding site in the middle, And so, as I said, heme is the molecule that causes the red color in blood. And it's also the molecule that facilitates binding of oxygen within myoglobin and hemoglobin. And so both myoglobin and hemoglobin both have this heme, what's called a cofactor, that's bound to the proteins. And so these are very similar binding sites in both of these proteins, but you'll notice the major difference between myoglobin and hemoglobin is myoglobin is a monomer. And so the molecular weight is about 16,700 Daltons, whereas hemoglobin is a tetramer. So recall when we talk about quaternary structure, this is what we're talking about. So we have several subunits. We have 4 subunits in hemoglobin. So alpha 1, alpha 2, beta 1, and beta 2 shown in the right-hand side. And so this is about 4 times as large of a protein as myoglobin is, with a molecular weight of almost 65,000 Daltons. And it's a tetramer. And so this is a oligomeric protein, exhibits quaternary structure, and that's actually really important for the different binding affinities, or the different O2 binding curves that are exhibited between myoglobin and hemoglobin. And so this is what we're going to look at in the next few lectures, is how does this structure affect these differences? And so we'll get to that starting now.